Hello, we all know that several proteinaceous enzymes require an additional chemical component called cofactor. Cofactor may be either inorganic ions or complex organic compounds. These complex organic compounds are known as coenzymes. Generally, coenzymes are derived from B vitamins. Such coenzymes are actively involved in the catalytic reactions of enzyme by serving as intermediate carriers of functional group in biochemical reactions. Moreover, deficiency of precursor vitamins of coenzyme leads to certain disease conditions. In this e-lesson, we will discuss about the structure, function, and reaction mechanism of coenzyme pyridoxal phosphate, folic acid coenzyme, B12 coenzyme, and we will also discuss about the deficiency diseases of pyridoxal, folic acid, and cobalamin, the precursor vitamins of these coenzymes. Now, what is coenzyme? We know a number of proteinaceous enzymes require an additional chemical component called cofactor other than their amino acid residues. Such cofactor containing enzyme is known as holoenzyme. The cofactor may be either inorganic ions such as iron, magnesium, manganese, or zinc, or a complex organic compound such as pyridoxal phosphate, tetrahydrofolate, 5' deoxyadenosylcobalamin, nicotinamide adenine dinucleotide, flavin adenine dinucleotide, thiamine pyrophosphate, lipoid, biocytin, coenzyme A, etc. These complex organic cofactor is called coenzyme. Usually, coenzyme is actively involved in the catalytic reaction of enzyme by serving as intermediate carrier of functional group in biochemical reactions. Enzymes that use same coenzyme usually perform catalysis by similar mechanism. Generally, such important biomolecule called coenzymes are derived from B vitamins. Now see, coenzyme pyridoxal phosphate transfer amino group, and it is derived from precursor vitamin pyridoxal, that is vitamin B6. Then tetrahydrofolate, a folic acid coenzyme, transfer aldehydes, and precursor vitamin is folic acid. B12 coenzyme 5' deoxyadenosylcobalamin transfer hydrogen atom and alkyne groups, and it is derived from cobalamin, vitamin B12. Now, coenzyme pyridoxal phosphate PLP. Pyridoxal phosphate is a coenzyme derived from vitamin B6, that is pyridoxal. This is a structure of coenzyme pyridoxal phosphate, and it is pyridoxal, vitamin B6, the precursor of this coenzyme pyridoxal phosphate. Then, the function of pyridoxal phosphate. Pyridoxal phosphate serves as a coenzyme for different enzymes catalyzing transamination, decarboxylation, and racemization reactions. Enzyme alanine aminotransferase, alanine racemase, glutamine decarboxylase are examples of pyridoxal phosphate containing enzymes. Then, the reaction mechanism. In this reaction, aminotransferase catalyzes the transfer of an alpha amino group from one alpha amino acid to alpha keto acid. This enzyme is also known as transaminase. This enzyme generally funnels alpha amino groups from a variety of amino acids to alpha keto glutarate. This is alanine aminotransferase enzyme catalyzed reaction. Here, amino group is transferred from alanine to alpha keto glutarate, and then pyruvate and glutamate are formed. All aminotransferase enzymes contain the prosthetic group pyridoxal phosphate. The most important functional group on pyridoxal phosphate is the aldehyde. This group forms covalent CYP-based intermediate with amino acid substrate or lysine residue of the enzyme. CYP-based intermediate with amino acid substrate is called external aldemine, and the date with the lysine residue of the enzyme is known as internal aldemine. The CYP-based butene, the amino acid substrate, and pyridoxal phosphate loses a proton from the alpha carbon atom of the amino acid to form a quinonoid intermediate. Reprotonation of this intermediate at the aldehyde carbon atom yields a catamain. The catamain is then hydrolyzed to an alpha keto acid and a pyridoxamine phosphate PMP. These steps constitute half of the transamination reaction. The second alpha keto acid reacts with the enzyme pyridoxamine phosphate complex to yield a second amino acid and regenerate the enzyme pyridoxine phosphate complex. The sum of these percent reactions is amino acid 1 and alpha keto acid 1 reacts to form amino acid 2 and alpha keto acid 2. Deficiency disease of pyridoxine phosphate. As coenzyme pyridoxine phosphate is important for many enzymes involved in the amino acid metabolism, glycogen metabolism, and also for steroid hormone action. Deficiency of pyridoxine, that is vitamin B6, the precursor vitamin of coenzyme pyridoxine phosphate, causes depression, confusion, convulsion, and hormone-dependent cancer of the breast, uterus, and prostate. However, excess pyridoxine causes sensory neuropathy. Next is the folic acid coenzyme. Tetrahydrofolate TSF is the folic acid coenzyme. It is derived from vitamin folic acid. This is the structure of coenzyme tetrahydrofolate and it is the precursor vitamin folic acid. This coenzyme consists of 3 groups: a substituted pyridine, para-aminobenzoate, and a chain of 1 or more glutamate residues. Function of tetrahydrofolate, the folic acid coenzyme. The coenzyme tetrahydrofolate and different tetrahydrofolate derivatives are highly versatile carrier of activated 1-carbon unit in a variety of biosynthetic and degradative reactions. Enzyme glycine synthase, serine hydroxymethane transferase, methionine synthase are examples of tetrahydrofolate containing enzymes. Next is the reaction mechanism. The 1-carbon group carried by tetrahydrofolate is bonded to its N5 or N10 nitrogen atom denoted by N5, N10 or to the bot. This is a general biochemical reaction of glycine synthesis catalyzed by serine hydroxymethane transferase. In this reaction, glycine is formed from the serine through the removal of 1-carbon group. Tetrahydrofolate accepts beta carbon C3 of the serine, which forms a methylene bridge between N5 and N10 to yield N5 and 10 methylene tetrahydrofolate. Then, deficiency disease. Deficiency of the folic acid, that is the precursor of the coenzyme tetrahydrofolate, or deficiency of vitamin B12, which leads to functional folic acid deficiency, affects cells that are rapidly dividing. Clinically, this affects the bone marrow and leads to megaloblastic anemia and neuron tube defects. Folic acid supplement reduces the risk of neuron tube defects and may also reduce the incidence of cardiovascular disease, as well as it also reduces the incidence of colorectal and other cancers. Next is B12 coenzyme. 5' deoxyadenosylcobalamin is a coenzyme derived from vitamin B12, that is cobalamin. It is also known as B12 coenzyme. This is a structure of coenzyme 5' deoxyadenosylcobalamin and it is the precursor vitamin B12 cobalamin. The core of cobalamin consists of a corin ring with a central cobalt atom. The cobalt atom is bonded to the 4 pyronitrogens. The 5th substituent link to the cobalt atom is a derivative of dimethyl benzimidazole. In coenzyme B12, the 6th substituent linked to the cobalt atom is the 5' deoxyadenosine unit. This position can also be occupied by a cyano or a methine group or other ligands. Then, the function of coenzyme 5' deoxyadenosylcobalamin. Cobalamin enzymes, that is enzymes containing B12 coenzymes, are present in most organisms and catalyze 3 types of reactions. Number 1, intramolecular rearrangement. Number 2, methylation. Number 3, reduction of ribonucleotides. In mammals, reactions like conversion of L-methylmalonyl-CoA into succinylCoA and formation of methionine by methylation of homocysteine require cobalamin enzymes. Next is the reaction mechanism. In this reaction, methylmalonyl-CoA mutase catalyzes a rearrangement reaction to form succinylCoA. The rearrangement reaction catalyzed by this cobalamin enzyme is exchange of two groups attached to adjacent carbon atoms. A hydrogen atom migrates from one carbon atom to the next, and the CO-CoA group of methylmalonyl-CoA concomitantly moves in the reverse direction. The first step in these intramolecular rearrangements is the cleavage of the carbon-cobalt bonds of 5' deoxyadenosylcobalamin to generate the CO2 plus form of coenzyme B12 and the 5' deoxyadenosyl radical. In this homolytic cleavage reaction, one electron of the cobalt carbon bond stays with cobalt, while the other stays with the carbon atom, generating a free radical. Then, the highly reactive 5' deoxyadenosyl radical abstracts a hydrogen atom from the substrate to form 5' deoxyadenosyl and a substrate radical. These substrate radicals spontaneously rearranges. The carbonines CoA group migrate to the position formerly occupied by hydrogen on the neighboring carbon atom to produce a different radical. This product radical abstracts a hydrogen atom from the methane group of 5' deoxyadenosyl to complete the rearrangement and return to the deoxyadenosine unit to the radical form. The role of coenzyme B12 in such intramolecular migration is to serve as a source of free radicals for the abstraction of hydrogen atoms. An essential property of coenzyme B12 is the weakness of this cobalt carbon bond, which is readily cleaved to generate a radical. Now, deficiency diseases. Vitamin B12 deficiency impairs the metabolism of folic acid and leads to functional folate deficiency. Its deficiency causes pernicious anemia and methylmalonic acidosis. The most common cause of pernicious anemia is the failure of the absorption of vitamin B12 rather than dietary deficiency. So, we can now conclude that there are numerous proteinaceous holoenzymes that require an additional chemical component, either inorganic ion or complex organic compound. Usually such complex organic compound known as coenzyme is actively involved in the catalytic reaction of the enzyme. It serves as an intermediate carrier of functional group. Coenzyme pyridoxal phosphate transfer, amino group, tetrahydrofolate are important for transferring activated one-carbon unit in a variety of biosynthetic reactions and degradative reactions. And coenzyme 5' deoxyadenosylcobalamin transfer hydrogen atoms and alkyne groups. Coenzyme pyridoxal phosphate, tetrahydrofolate, and 5' deoxyadenosylcobalamin are derived from pyridoxal, folic acid, cobalamin. So deficiency of such precursor vitamins disturb reaction catalyzed by coenzyme dependent enzyme and consequently leads to various disease conditions. Thank you.